What are operons? Good question. An operon is basically a cluster of genes controlled by an operator. Something called a repressor binds to the operator, which then stops or starts the transcription of multiple genes. In other words, operons control the flow of RNA polymerase. This may sound like a bunch of jargon, but we'll give you two examples that will hopefully help clear things up. The first is called a repressible operon.
This is a TRP operon. It is tasked with tryptophan synthesis. It is a repressible operon, which means that it is an operon where transcription is on. It is stimulated to turn off by a protein called the repressor. A gene called the regulatory gene produces this protein you see here. It is an inactive TRP repressor, which means that it is going to turn off the operon and block transcription. The smaller part is a tryptophan, a co-repressor. It binds to the repressor and activates it. The two make their way to the promoter, where RNA polymerase binds and transcription starts. Next to the promoter is the operator, which is where the repressor will bind. Once it fits in, the RNA polymerase is blocked from transcription and the operon is officially turned off.
This previous example is a kind of gene regulation called negative control. This means that the operon is switched off by an active form of a repressor protein. The next example is going to be positive control, in which the regulatory protein interacts with the genome to increase transcription. This example involves a protein called cAMP and CRP. Here we start with a lactose gene. When glucose is scarce, a receptor protein called cAMP is created. It then binds to a protein called CRP, which then activates it. The now active CRP protein goes upstream and binds to the DNA promoter. This is to increase transcription and increase production of polymerase. As such, the CRP protein is a promoter. It is an activator that causes more RNA polymerase to come. There is no repressor whatsoever in this example. And that's about it. Thank you for listening and watching, and we hope that this helped to clarify things.